Chapter 11 is talking about hearing and balance. For hearing and balance, ear is responsible for it. So there is outer ear, middle ear, and inner ear. For outer ear, you can see there is a structure that you can see it from outside. Is visible to anybody. That's called auricle. This area of the external ear or outer ear is called concha. Concha is connected to the external auditory meatus, which is a large opening, and from here to here is the external auditory canal. So this is meatus, this area, and the canal is the whole from here to here. External auditory canal is separated from the middle ear by this structure, and this is called the tympanic membrane. The middle ear is a cavity. It contains some ossicles. Ossicle means small bones. So these are three ossicles here: malleus, incus, and stapes. Malleus is connected to the tympanic membrane. Incus is connecting malleus with stapes, and stapes is connected to an area is called oval window, and that is oval window is separating inner ear from the middle ear. The middle ear cavity is also connected to pharynx, so this is called pharyngotympanic tube or eustachian tube. And sometimes this area is blocked, and this is why when you travel there is a change in the pressure, and this is why you get ear pain. And if you chew something or if you yawn something, this area opens up and the pressure on both sides balance out, and the pain usually decrease. There is another area, this is called the round window. This thing that you see, this is also an area that's separating the middle ear from the inner ear. So the inner ear is made of two parts. One is called Cochlear, and this is responsible for hearing, and another area that is for balance. So for balance there is two different areas. One is called the vestibule. This is just the spelling of the vestibule. It was not in your textbook, so just write it down. Vestibule. You can put an arrow there. And the other part is called semicircular canals. So once again, semicircular canals, which is this area, and the vestibule, they are both part of the inner ear, which is responsible for balance. The other part of the ear, inner ear, is called cochlea or cochlea. Both of them are correct pronunciation, and that is for hearing. So now it's talking about some of the disorders that are affecting the outer ear and middle ear. One is obstruction. So for obstruction, usually it can be a foreign body. Wax is although the most common cause, and when people are cleaning their ear, they are trying cleaning it. Some people are pushing the wax more deeply, and that's why it creates it makes the wax harder, and that's why it blocks the external auditory canal and makes the hearing more difficult. This is easy to treat, so just remove the wax or remove the foreign body. It's easy to treat. Another condition is called otosclerosis, and in the otosclerosis it's a progressive hearing loss. This is the cause is not known. It happens in young to middle-aged adults, and the ossicles fuse together. There's a lot of fibrosis formed there, and the person cannot hear anything. So it is it's a common cause of the progressive hearing loss. The obstruction usually happens faster, but this is a progressive hearing loss. And otitis media is common in young children, so usually between age 1 and 3. This is very, very common. Some of you may have had that. So this can be a viral infection, or it can be a bacterial infection. Bacterial infection is, of course, more serious, and pus forms inside the middle ear that may cause meningitis, or it may cause brain abscess. Both of them are very, very serious conditions. And for viral infection, you just have to wait, and it goes away. You can give some medication for pain, but for the bacterial otitis media, you have to give antibiotics because you want to prevent complications. Sometimes the tympanic membrane ruptures and the pus is coming out of the ear, so it's a common condition. If a child is crying a lot, is very irritated, and pulling the auricle upward, the area is red behind the ear, so that's likely the person may have otitis media. So now let's talk about hearing. For hearing, is cochlea is involved. If you cut the cochlea, you can get something like this. So this whole area that you can see where the arrow is pointing to, that is called bony labyrinth. Bony labyrinth. So this is like a wall made of bones, and this is the part of the tympanic bone inside the tympanic bone. I mean temporal bone. Sorry, temporal bone, and that is wall of the cochlea. So another structure is called membranous labyrinth. So this is the membranous labyrinth. If you look at it, the bony labyrinth is made of the vestibular duct and the tympanic duct, and the membranous labyrinth is made of the cochlear duct. The membranous labyrinth is separated from the tympanic duct by the basilar membrane, and on top of the basilar membrane, inside the cochlear duct, you have hair cells, and that is connected to a nerve, and the cell bodies of those nerve fibers are located in the spiral ganglia. The image below, if you look at it, so this is the start of the oval window. So this is the stapes. Stapes should be here. So this is a little bit mistake in this image. Stapes should be here, and the oval window is separating the middle ear from the inner ear. So the beginning of the vestibular duct is starting from the oval window, and there is a fluid there, and that fluid is called perilymph. It goes all the way to the tip of the cochlea, so this area, and that is called helicotrema, and then it continues with the tympanic duct. Tympanic duct also contains perilymph, and then the tympanic duct ends with a round window, and round window is this area, and that separates the inner ear from the middle ear, which is around this area. This is the histology of the bony and membranous labyrinth. So this area, this whole area, as you can see, is the bone. This is part of the cochlea, and this is the vestibular duct, starting from the oval window, and contains perilymph. Then it goes all the way to the apex, and then it changes and becomes the tympanic duct, and this also contains the perilymph. In the middle you have the cochlear duct, and you can see these two membranes. These are made of connective tissue and epithelial tissue, so this is why it's called membranous labyrinth. This area contains endolymph, which is a little bit different here. Hair cells are located here in the basilar membrane, and these are the nerve fibers, and it goes all the way to here, and the cell bodies are in the spiral ganglia. So spiral ganglion contains afferent cell bodies that are coming from the hair cells in the basilar membrane. By the way, one more thing that I should have said in the previous slide is are these three names that I wrote it here. It's not in your textbook, so it's just a different name. Sometimes if you read it in a different source, you can have these names. So scala vestibuli is similar to the vestibular duct, scala tympani is similar to the tympanic duct, and scala media is similar to the cochlear duct. It's just a different names that if you read it in a different source, you may come to this kind of term. So it's just same name, same meaning. Okay, so this shows a little bit higher zoom of the cochlear duct here. This area is called hysteria vascularis, so it's very vascular. A lot of blood vessels are there, and the potassium is leaving the blood and is concentrating in the endolymph. So endolymph contains a lot of potassium. This membrane that you see here, this is called the Reisner membrane. Another more common name for that is called vestibular membrane, and that is separating the vestibular duct from the cochlear duct. This area that you see, this is the basilar membrane. So the basilar membrane is separating the tympanic duct from the cochlear duct. And the basilar membrane contains hair cells. So this is the hair cells. These are called outer hair cells, and this is important for amplification and fine-tuning of different sounds. And this is the inner hair cells, and this is important for processing sound. So if the sound is lower, then the outer hair cells can amplify that sound. And this is the tectorial membrane. I have a different image for that, and that you can see later, and that is embedded. The outer hair cells are embedded in the tectorial membrane. Nerve fibers are here, and from the outer and inner hair cells, and then goes through, and that creates part of the cranial nerve 8. So cranial nerve 8 has two divisions. One is the cochlear division, that is for hearing, and the other one is the vestibular division, which is for balance. So this is how the sound transmits and is processed. So auricle is responsible for picking up the sound and concentrating it and putting it into the external auditory canal, and then when the sounds here is highly concentrated and it hits the tympanic membrane, it moves the tympanic membrane, and this movement will cause what? Cause movement of these three ossicles. So malleus first, incus, and then stapes. So because malleus is bigger and then the stapes is smaller, so that will cause amplification of the sound. The stapes is connected to the oval window. So this image is actually the correct image. The couple of slides ago that was incorrect, because it shows showing stapes is here but is not there. So stapes should be in the beginning of the vestibular duct. So vestibular duct contains fluid. Now when the sound comes, the stapes pushes in, and when it pushes in, it moves the fluid, so increase the pressure here in the vestibular duct. As the pressure increases in the vestibular duct, then depends on the frequency of the sound, the cochlear duct also moves, and that will also cause changes in the movement of fluid in the tympanic duct, because they are connected. As you can see here, the vestibular duct is connected to the tympanic duct, so that's why when a higher pressure here, then that pressure goes to this area. Pressure always goes from higher area to the lower area, so that's the pressure gradient. So when the pressure comes and the fluid moves this way, the fluid waves, it will push the round window. So now the round window bulges into the middle ear. So once again, when you hear a sound, the oval window pushes into the vestibular duct, fluid moves, and when the fluid comes back, now the round window bulges into the middle ear cavity. There is the fluid does not drain there, it's just bulging out. And you can also see that you can have the cochlear duct, and the cochlear duct contains organ of Corti, which we will talk about it. So this is the area that hair cells are located, and then you can see these nerve fibers are located and coming from those hair cells in the cochlear duct, and it goes to the spiral ganglia. So if there's a high frequency sound, usually these areas are stimulated. So these areas of the cochlear duct and the hair cells are stimulated. And if it is a lower frequency, usually the tip of the apex is stimulated, so the nerve fibers here are stimulated. So this is the organ of Corti. So you can see this is the cochlear duct, so made filled with endolymph that contains a lot of potassium, and the cochlear duct is separated from the vestibular duct by the vestibular membrane here, and the cochlear duct is separated from the tympanic duct by the basilar membrane. On top of the basal membrane you have these hair cells, and these are the supporting cells. There's two types of hair cells. One is the outer hair cells, and the other one is inner hair cells. The inner hair cells do not directly connect with the tectorial membrane, but the outer hair cells are close in contact with the tectorial membrane. So when the fluid moves and it comes through the tympanic duct, this basilar membrane goes up and down, and these hair cells stimulated by the tectorial membrane, and then is stimulating the nerve fibers. And the fluid also moves in the cochlear duct, so that's why the inner hair cells are also stimulated and send signal to the brainstem through the spiral ganglion. This image, you can see that the nerve fibers, majority of the nerve fibers are leaving the hair cells, and these are the afferent cells, and that makes the majority of the cochlear nerve. So many of those nerve fibers are coming from inner hair cells, some of them are coming from the outer hair cells, and these are afferent, so this is for processing sound. But there is also another mechanism. These are the efferent ones that are going to the outer hair cells, and they are inhibiting the response of the outer hair cells when the fluid, when waves comes, the basilar membrane moves, and also the tectorial membrane moves. So that reduces the amplification of the sounds that happens by the outer hair cells. So majority of the cochlear nerve contains afferent nerves toward the brainstem, but some of them are coming from brainstem. So this slide shows the physiology of hair cells. These are specialized epithelial cells, and they have microvilli. These are called stereocilia. This is a spelling. Stereocilium is a singular form. Stereocilia is the plural form. Kinocilium is the longest of those microvilli, and you can see these proteins that are connecting the tips of these stereocilia. In the tips you can see that potassium channels are there. So normally if there is a movement of the kinocilium toward the rest of the stereocilia, nothing will happen. So this is the hyperpolarization. But if these stereocilium moves toward the kinocilium, then the tip links proteins are stretched out. When they are stretched out, then because they are attached to the potassium channels, they open up the potassium channels, and inside the cochlear duct there's a lot of potassium there. So these potassium ions enter the stereocilia and enters the hair cells. So potassium is positively charged, and that will cause depolarization. Normally sodium enters the cell, and that will cause depolarization. Potassium leaves the cell, but this is an exception there. Because of the depolarization, calciums enter now the cell, and if you remember, calcium is important for fusion of the vesicles to the plasma membrane of the cells, and then these vesicles contains glutamate, that's the neurotransmitter, and that will cause exocytosis, and the postsynaptic membrane here is picking up, and then it will cause activation of the nerves here, and then that's how a person hears sound. Something that is missing in your textbook, you can add this if you want. So this is a spiral ganglion. That is the one that the cell bodies of these afferent nerves are located there, and that is coming from the hair cells. Cell bodies here, and then that is in the cochlea. So then the fibers are going to the brainstem. So this is the cochlear nuclei are located. This is the location of the cochlear nuclei. The cochlear nuclei is located in the rostral medulla and in the caudal pons. So this is the pons. So this is the area that is, the cochlear nuclei are located, and that process the information, and then it crosses the midline and goes to the inferior colliculus. So the fibers here are called lateral lemniscus, and if you guys remember, there was also medial lemniscus, and that was responsible for movement of the eye. So this lateral lemniscus, it goes to the inferior colliculus. Inferior colliculus, if you remember, it was in the midbrain. So from there it goes to the medial geniculate nucleus, and the fibers here is called inferior brachium. The medial geniculate nucleus is located in the thalamus. From there it goes to the transverse temporal gyrus in the cortex of brain, and the information is processed there. And depends on the frequency of the sound, it goes to different areas. It's not shown here, but it goes to different areas in the transverse temporal gyrus. So some causes of hearing loss. So this is also called sensorineural hearing loss. There can be drugs and toxins, so quinine, aspirin, and streptomycin, some of them. Prolonged exposure to the loud noise, for example, hearing to my lecture, that can be a prolonged exposure. And stuff that you can listen to Corti songs, so she can sing better, so that will not cause any damage to your hearing sensation. Infections like rubella and during pregnancy, cytomegalovirus and syphilis can cause nerve deafness. Probably the most common cause of the nerve deafness is called presbycusis, and that is happening with age. So as the person gets older, so they lose some of the hair cells, and that will cause nerve deafness. And also tumor, which is not that common, and that is the tumor, benign tumor of Schwann cells, and that will cause unilateral deafness, tinnitus. It may compress the facial nerve, because the facial nerve also passes through very close to that, and they may also get some muscle weakness on the face. Sometimes they get pain in that area, so it sometimes for diagnostic purposes it's a little bit difficult. You don't know, is it acoustic neuroma, or is it a problem from the cranial nerve 7, so you have to do some MRI to know which one it is. So the second part of the inner ear is the balance. This is the cross section of the inner ear. So you can see this area, we already talked about it, and this is the cochlea. This is the tip of the cochlea, so helicotrema, and these are the different ducts inside the cochlea. So you can, starting from here, this is the stapes and this is the oval window, and this goes like this, and that is the vestibular duct, and goes all the way to the tip and comes back like that, and this is the tympanic duct and ends with the round window. So see, you can see both of them are separating the middle ear from the inner ear, and this green area that you see, that is the cochlear duct, and this is where the hair cells are located. But we already talked about this, so let's forget about this area. So this is the part that is responsible for balance, and I can separate that into two different areas. One is the semicircular canals. Okay, so this area is the semicircular canals, but you can see there is two different names there. One is a canal and the other one is the duct. So the canal is actually the bony part. So these are the walls. So this is a bony, like a bony labyrinth, and the duct, you can see these green areas. These are the membranous labyrinth. So this is the one. That's why they have two different names there. So ducts are located inside canals. So and the ducts contains the endolymph. Okay, and the canal contains perilymph. So there is three different semicircular canals are there. So this is the anterior one or superior one, and this is the one that is the horizontal one or transverse one, and this is the posterior one. You can see at the end there is an enlarged area, and that's called ampulla. Okay, some hair cells are located. So this is light green, these are the hair cells. So hair cells are located inside ampulla. Ampulla is connected to this whole area that's called vestibule. Vestibule has two different areas. One is called utricle. Utricle is close to the semicircular canals. Okay, so these ampullae that you see, ampullae is the plural form of ampulla. So these are connected to the utricle, and there is another one that's called saccule, and saccule is closer to the cochlea. These utricle and saccule, these two parts of the vestibule are connected to each other, and that is the utrico-saccular duct. These two ducts combine, and that makes the endolymphatic duct. And this thing that you see, this enlargement, this is the endolymphatic sac, and then this will drain in the subarachnoid space. So let's talk about the semicircular canals, which is important for movement of the head in different rotation, different areas. You can see again that there is different semicircular canals. Inside should be the ducts, not shown here properly, and then these are enlarged areas, and these are the ampullae. This area that's enlarged. Inside this ampullae, you can see something else. This is called the crista ampullaris. Okay, so this is the crista ampullaris. This area is, this is called crista ampullaris, which is around here. This area. So these are made of the simple columnar epithelial tissue, and then these are the supporting cells, and you can see these hair cells here. This thing that you see, this is called cupula, and this is made of gelatinous protein, and it's floating in this fluid, and that is the endolymph fluid that is inside the semicircular ducts here. Okay, somewhere here. So this is the fluid there, and you can see that there's microvilli or stereocilia that you see here that is embedded in the cupula. Then the hair cells are connected to the nerve, and then any time there is a changes here, then the nerve senses it and then sends it to the brainstem. So this is a you know, if somebody moves his head in different directions, three different planes of directions, and here is an example of that. So if somebody is moving, I said, from right to left, then you have changes that you can see in the semicircular canals here. So you have it, the semicircular canals on the left side, and you have this on the right side. So when you see it here, when you move your head, the fluid in the semicircular canals moves in the opposite direction. So that moves in the opposite direction. So move your head, and that will cause an opposite direction here. So when it depends on these hair cells that you can see here. So when the fluid comes there, then this stereocilia move toward the kinocilium, and you can see that these tip links proteins are there. That opening, that potassium channels there, and that will cause a depolarization. So if you are moving your head from right to left, and that will cause movement of the fluid to the opposite direction, and that will cause a depolarization. What happens to the other side? So you move still, your head is still moving to the left, but the fluid is moving to the right side. So this is an opposite side, and now the stereocilium is the kinocilium actually in this case moves toward the stereocilia, and there's no opening of the potassium channels because there's no opening there. So potassium does not enter the cell, and there's nothing happens here. So there's no depolarization. So that will hyperpolarize, which means that there is not much information is processed. There is no action potential there, so nothing will happen there. And that way you feel that movement on an area that caused depolarization. The vestibule also have, so this is the vestibule, so the utricle and saccule, as I talked about it before, they also have hair cells, and these are called macula. So the macula is located in the utricle and saccule. So these hair cells are there, and it has a stereocilia, but it has something else. So this is a membrane. Okay, it's a gelatinous membrane. This is called gelatinous membrane. And there is also have calcium carbonate on top of that, and that calcium carbonate is called otolith. So like small stones of the ear. So if you translate it, otolith, and they are embedded on top of this gelatinous membrane, and depends on the movement of the head, then the effect of gravity, it will move these calcium carbonates, and this is how these hair cells are activated, and then it will cause activation of the nerve fibers, and then you can feel the different direction. So for saccule is important for movement of the head up and going to a vertical plane, up and down, for example going in an elevator, or moving the head anterior to posterior. And utricle is moving the head left to right, or in a lateral displacement from left to right, sideways, going left to right. So both of them are important for linear acceleration. For the comparing this with semicircular canals and ducts, that was for rotational movements, and this is for linear acceleration and deceleration. For the vestibular pathways, there are three different reflexes are there. One is called the vestibular ocular reflex. This is important that when you move your head, also the eyes are moving, so that way the object can stay on top of your retina and the fovea, and we talked about this in before chapter 9. So that movement is for that, so you can see things clearly. The second one is called vestibular cervical, and this is connecting the nerve fibers to the cervical area and the muscles that are supplying the neck area, and it's important for adjustment of the head and neck movements. And vestibular spinal reflex, that is going to the torso muscles and also the proximal girdle muscle, girdle, Girdle, shoulder and hip and girdle muscles, and that's also for posture and also for tone of those muscles. So you have these vestibular nuclei. There is two of them. One is the medial one and the other one is the lateral one. The medial one is important as processing information for the rotational acceleration, that's getting information from semicircular canals. Some of them are going to the muscles in the neck area, and some other ones are going all the way down and then the torso and in the girdle area. So the pathway for that is the medial vestibulospinal tract, also the MLF. So there is a ascending MLF and also descending MLF. This is a descending one. If you go back to the brainstem, you can see the MLF in that brainstem section also. And the lateral vestibular nuclei, these are for the linear acceleration. So this is getting information from the vestibules, from utricle and saccules, and that information is going to the limbs. So these are for extensors of the limb. So they're going in a different area, and the pathway for that is called the lateral vestibulospinal tract. If you remember from chapter 8, lateral vestibulospinal tract is on the same side, ipsilateral, does not cross it, and the medial vestibulospinal tract is bilateral on both sides. The diseases of the balance, there is 3 of them that are common. So I put it here. One is Meniere disease, the other one is labyrinthitis, and the third one is the benign positional vertigo. It's a different. I want to tell you it's a little bit easier way to remember all of these things. In Meniere disease, the cause is not known. Nobody knows it. There are different risk factors. There are different, factors that may increase the possibility of getting Meniere disease, but the actual cause is not known. Labyrinthitis, it's usually an infection. Viral infection is more common. Also, some toxins like alcohol, quinine, and salicylate like aspirin can cause labyrinthitis. And benign positional vertigo is those otoliths that I talked about it before. In some people, those otoliths dislodge and they go to semicircular canal. So when they move their head, this otolith moves in a different direction, and that will stimulate the hair cells in the crista ampularis. So if they don't move their head, they do not get vertigo. So that's the cause. From sign and symptom point of view, all 3 of them, they will get vertigo. Okay, all 3 of them have vertigo, and they also get nystagmus, which is the movement of the eyes. But with Meniere disease and labyrinthitis, they also get hearing loss and nausea and vomiting, and sometimes the fullness of the ear and some pain in the ear. So how can you differentiate the Meniere disease from labyrinthitis? If the symptoms and signs are the same, then you have to rule out labyrinthitis. You have to take a good history, so the person, you know what kind of medication they took, if they were exposed to any kind of toxins, if they have any kind of infections or not. So once you remove and rule out all those causes, then you can say, okay, the person may have Meniere disease. Benign positional vertigo, the main symptom is the vertigo and dizziness. And that's associated with movement of the head. So that way it's a little bit easier to diagnose. And that was the last slide.